All right, section 19 is going to talk about the Hardy-Weinberg theorem. So let's talk about this for a minute. So the Hardy-Weinberg principle or theorem was developed by two gentlemen, Godfrey Hardy, which was an English mathematician, and Wilhelm Weinberg, which was a German doctor. Each of them worked alone, and they came up with some fundamental principles of population genetics. Now today, we know these principles as the Hardy-Weinberg theorem or principle. This shows how allele frequencies do not change in a population if certain conditions are not met. So let's talk about those conditions and what we have to do. So in this principle, it used non-evolutionary populations. These are ones that didn't change. So that's who we're describing. The shuffling of alleles. Now remember, alleles, if we have a trait, we have two alleles for every trait. Such as if you have a black cat, it might be big B, big B. Each of those B's would be an allele. Now, the shuffling of alleles by meiosis, which is producing sex cells, would be random fertilization and no effect over the overall gene pool. So you're not getting any crazy mutations occurring like that. Now, the natural populations are not expected at expected to any real change in this. So your natural populations really aren't going to be too affected by this. Now, there is some deviation that will usually result in evolution, and understanding that non-evolutionary populations help to better understand why evolution occurs and how it occurs. So there are 5 basic assumptions when you do the Hardy-Weinberg or HW principle. 1, you have to have a large population size. Small populations don't have as many alleles, so you don't have that fluctuation alleles, so you don't really see a big change in that. No migration. The immigration can change the frequency of alleles by bringing in new alleles or new genetic information into the population, and so we're not taking that in. 3, there's no net mutations. And so for this, all the alleles change from one to another. We don't have any big mutations that come into the population that would really affect something. Random mating. You would select traits randomly or your mates randomly. There's no selection for a specific trait in there. You're not looking for the biggest mane for the lion. You're actually just randomly selecting your mates. And no natural selection. So this means every species has the same opportunity to survive with this. And there's an equal chance of reproducing and passing those genes on to the next population. So if we look at all these things, like usually with the bird, he's going to puff up his chest to get a mate. That's not going to happen. You're not going to show those little mating dances in there. You're not going to have the nice coloring for the pheasant or the larger horns so that you could fight off other bucks to become the dominant male within the population that breeds all the species. And with the lion, it doesn't matter if you have any, if you're the larger male with a bigger mane and showing those, you're not going to have those. So these males here, their physical characteristics would not be important according to this principle, which is just, it's really not true. These are important characteristics, but we look at this principle because it actually looked at conditions on how we could actually see genes being expressed and genotypes becoming expressed within a population. So here we go. So with this principle, we have the gene pool for a non-evolving population. That means it's not changing, remains constant, and we have a theorem that goes with this, or an equation. P is going to be the frequency of our homozygous dominant genotype. Our PQ is going to be our heterozygous genotype, and our Q is going to represent our homozygous recessive. Now remember, the dominant alleles will always show up over the recessive alleles. And so we're going to see that capital A will be expressed over the lowercase a. And because we can look at this, we can kind of see the frequency at which it occurs in a population. Now with this, we can see that P and Q added together should always equal 1. And so here's going to be kind of a breakdown of what we have. So if we have 500 flowering plants, if we have a breakdown of 480 of them with red flowers, 20 of them with white flowers, here's how it's going to break up. That recessive trait, which is going to be our white flowers, is only going to be found in 20 plants, because remember, it won't show up unless it's there. And then our dominant trait is going to be broken up into heterozygous dominant and homozygous dominant traits. And so we can kind of see the breakdown. I do have an attached video that's going to specifically go through this portion of it, so it's a little bit easier for you. And we can see how this breaks down in the population, and we can have an overall R factor, which is what we're looking for. And so we can see how much of that population is actually going through the gene pool and the probability of it getting passed on throughout the gene pool. Granted, all genes are being shown here. We're seeing a genetic variation. Now, our 3-2-1 phenotype is going to be expressed. Or sorry, our 3-1 phenotype is going to be expressed, and our 1-2-1 genotype is going to be expressed when we're mating two homozygous heterozygous parents together. So that understanding of genetics that we had from last year bio, that's still going to be into play here. So you can watch that supplementary video right now so that you can better understand this principle.